Oh my god. She runs away.
Sorry, what's your lesson?
Correct.
Scanning stuff, do you guys have papers? Kenneth, do you have a paper? Do you have a paper?
Are we supposed to show you?
I think it's always a good idea just to make sure that it's something that you understand, but that's your choice. You can pick the one that was originally assigned, or you can pick one.
Whoa,
that
you changed it up.
Just like we just when you're at it.
Yeah, no, I'm so sick. So,
I can't, we can't tell it. It's like,
no, I think it's just, yeah. And then when we off of your piece, and I know how much about it.
how long have you been on?
Since Monday. But he usually says that it usually is 5 days. With the rest of the process, it's supposed to be like 2 days.
I think it's
I like it, it's like,
You won't be able to test.
oh my god, I was up, like, I was like, he was like, so I just said, you know,
Yeah, okay, good. That's next one.
just fun, I know, me too. Yeah.
oh, we were wanting to go for a while, but then, finally decided.
Yeah. And then basically, I never questioned it after that.
It's pretty fast.
This one's delta, right? I know.
Okay, hi everyone. We're gonna get going if you don't mind. Okay, if you don't mind, we're gonna get going. So we're gonna try streaming this one lecture. We'll see how it goes, and we'll see what attendance looks like today, and then we'll make up our mind about whether the next couple will also be streamed or not. I apologize in advance. This should be lecture 5, and it is the muscular system as well as the cardiac muscle system. So your next term test is actually not that far off. It's in 2 weeks. It's on May the 30th, I believe. And that means that this lecture and the lecture that we have on Wednesday, next Wednesday, Monday is Victoria Day, are the only 2 lectures that you're going to have on your next term test. So in theory, you should be able to do really well because there's a very small amount of material, relatively speaking. But we do have a lot of material to get through, and it's really quite dense. The other thing I'm going to ask you to do is, if you haven't picked out a paper yet, is to go ahead and pick a paper out, start reviewing it, starting to look at other literature surrounding it. I'm told by Dr. Agard in the other section that usually the average on that assignment is somewhere in the B to B plus range, which is something that you can do and improve on simply by getting started a little bit earlier. So I would highly recommend that you do so. Again, we have 2 different systems that we're going to go through today, one being the muscle, and a lot of you will have already had muscle systems, skeletal muscle, smooth muscle, and cardiac. We're going to go through these really quickly. I'll ask you to know a few things in particular about these different systems, and then we'll come back and talk at the very end about the cardiac muscle system that you'll be continuing on with, as well as the respiratory system next Wednesday. So again, don't worry about all the details on here. Most of this is going to be covered in more detail on the subsequent slides. This is really telling you the 3 different muscle types that we're going to be spending time with. When we go through the different systems, for the skeletal muscle system, it's more important that you know all of the different molecules and how they work and the order in which they work as we go through the skeletal system. The smooth muscle system uses the exact same molecules, actin, myosin, calcium, but there are 2 different types of smooth muscles, and we'll talk about how these 2 different smooth muscle types, how they contract and how they're different when we start going through them. They're found obviously in different places. We don't control smooth muscle, skeletal muscle, we do, nor do we have control over cardiac muscle that we find only in one place, and that's in the heart. Again, this is also involuntary. It's controlled by a number of factors that we've looked at, like the endocrine system, as well as the autonomic nervous system that we spent some time on in the last few lectures. This is a slide that you should go back and use as a review slide. This is not a slide that I'm going to take specific data or information off of, but by the end of today's lecture, this is a really good review slide for you to come back and review the features of skeletal versus smooth versus the cardiac muscle type. And you can see that there are similarities as well as some differences between the different muscle types. And I'm going to go through them in more detail. It's really there for you to go back and review quickly. If you need a review page, this is a really good one to compare the different muscle types. So when we talk about muscle in general, so whether we're talking about the heart, whether we're talking about smooth muscle or skeletal muscle, really we're talking about how well it contracts. And when it starts contracting, how much force the contraction actually generates. Muscle doesn't contract by itself. It has to be stimulated to be able to contract, and that means that we have to excite it, and that means that we have to use the nervous system to excite it in order for it to contract and generate tension. Muscle also has this ability to stretch. So when you put a weight on your arm, that muscle for that split second actually stretches a little bit, and muscle has this property of extending or being able, and that's another general property, whether you're talking about the gut or the heart. Every time you think about it, the heart is filling, it is stretching as it fills with blood before it contracts again. Your skeletal muscles can stretch before they contract. And it's the same thing with your gut. Before it actually contracts, it actually stretches when you put food through it. So this extensibility is another common factor for all 3 muscle types. And related to this is the elastic property of the different types of muscle. Sometimes it's a little bit higher, so we have more elasticity in smooth muscle than we do in skeletal muscle, and cardiac muscle is always sort of in between. Okay, so if we look at the overall anatomy of skeletal muscle, and we're going to start with skeletal muscle, and we're going to spend some time going over skeletal muscle first, then we'll go through smooth muscle, and then eventually we'll get into we're actually going to see all different cell types here, but for the most part, we're going to be concentrating on this. What do you know what the other name for skeletal muscle is? So skeletal muscle, it controls the way our skeletons move, and we control it. It's voluntary. What's the other name for skeletal muscle? Any ideas? Have you heard it in any other courses? Another really common name for skeletal muscle. What does this look like? Yeah, it's called striated muscle. And when you look at skeletal muscle, it looks striated because there are regular bands of actin and myosin repeated over and over again. And you can actually see this at fairly low power under a microscope, that skeletal muscle has these striations. And we're going to be spending time trying to figure out what these striations are. When muscle contracts, actually move closer together as the muscle tends to generate force. These muscle cells that you see in here are comprised of this nucleus, and this is the actual muscle cell itself. This is one of these muscle cells. These muscle cells have all developed from myoblasts, and I'd like you to make sure that you are aware that they've developed from myoblasts. Overall, through development, and as you get older, up until a certain age, the number of myoblasts that you have remains relatively constant. Okay, and again, this striation, the light-dark patterns that you see here, these striations in skeletal muscle are things that we're going to try to figure out. So if we were to take one of those muscle fibers, so this is like one group or one muscle fiber, here's another muscle fiber that you see here, we're going to actually see that if you are talking about an actual muscle group, like in your arm or in your leg, it's comprised of many of these individual muscle fibers that I just showed you on the previous diagram. And it consists of a number of different layers. There is an outer layer that surrounds the entire muscle, known as the epimysium. And then there is the perimysium that surrounds individual muscle groups, or these individual fascicles that you see here. So here's like one group of muscle fibers. It would be surrounded by a perimysium. And then there is an inner layer known as the endomysium that surrounds individual fibers. So outside all of this, we have this muscular fascia, which is a connective tissue sheath. And do you guys know why you have connective tissue? You have muscle, and that's what we're going to be talking about. Why do you actually have connective tissue? What do you think it actually does? What does the name sound like it does? It connects things, right? In this case, it allows for the muscle to be connected with the blood supply, and this connective tissue makes sure that it all stays in place. And so this is there to hold all of these different functional groups, including the separate muscle fibers that are comprised of these connective tissue sheaths around them, as well as the capillaries and blood supply in the proper orientation. The other thing that we need to keep in the proper orientation is, since we are talking about skeletal muscle, and in order for it to contract voluntarily, you have to think about that muscle group contracting. The only way that we can do this is to activate these groups of motor neurons, and these motor neurons stimulate these muscle fibers. So that will be another big get to it that I need you to be able to understand. So right now we're looking at the overview of the muscle, how it's arranged, some of the anatomy of the muscle, and that we have this connective tissue sheath around the outside to keep the blood vessels in place. We haven't really talked about why we have striations. You'll need to know that when we get to it. How those striations contract, you'll need to know that when we get to it. And then how these motor neurons actually begin this whole process of causing your muscles to contract, because this is under voluntary control. That junction that we normally call a synapse between two different neurons, in this case, there isn't another neuron. It's a neuron, a motor neuron going to a muscle. We call it a neuromuscular junction. But in reality, it's really the same thing. It is a synapse. It is a connection between a nerve, and that the cell happens to be a muscle, and we call this a neuromuscular junction. Okay, is that okay for everyone? And I need you to be able to tell me later how a muscle contracts when you get an action potential from this motor neuron that comes in and activates it. So we just took a really quick look on that previous diagram about 2 slides ago about this muscle fiber. We saw that there were several nuclei. Those nuclei get pushed out to the edges. And this whole structure is actually, the membrane is called a sarcolemma. So when you see sarcolemma, it just means that the muscle, the plasma membrane, instead of calling it the plasma membrane of the muscle cell, we call it the sarcolemma. And you'll see sarcolemma when we come back to talk about action potentials a little bit later. The cells are also packed with all of these different myofibrils, and you'll need to know the 2 different protein, well, not 2, but the 2 major proteins, actin and myosin, that comprise these myofibrils, and we'll talk a lot about that starting with the next slide. And surrounding that, surrounding all these fibers, calling it the cytoplasm, we call this the sarcoplasm. It's the same idea, it's cytoplasm, but in this case, we call it the sarcoplasm. So we have all of these myofibrils that consist of thin filaments, which are made up of actin, and then we have also these thick filaments, which are called myosin. Together, actin and myosin make up these myofibrils. Okay. When we start building the muscle, the reason why we have these striations is that these actin and myosin filaments are sitting together like this, and they're sitting together like these 2 hands are repeated over and over again. And that these 2 hands that are joined like this right now, so actin, myosin, and it's going like this, the ends of this is called a sarcomere. This is one functional unit. And you have to imagine that this is repeated over and over again, and this is the idea of a sarcomere. And it's really important that you understand this concept. It's like the basic unit of what makes up a muscle cell. Any questions or things that I can clear up for you before we go on? Okay, so the basic unit of any muscle fiber that makes up any muscle is this sarcomere. Sarcomeres shorten, and we're going to understand how sarcomeres shorten in a moment. So this is that basic unit that I just mentioned to you. This is the sarcomere. It consists of the myofibrils. 2 myofibril proteins are shown here. We have actin and we have myosin. So we call this the thin filament, the actin filament, and this is the thick filament, or the myosin filament that you see here. This is repeated over and over again within this myofibril. This myofibril is one part of a muscle fascicle, and this muscle fascicle is one part of your muscle. So think of it repeating over and over again in one group, and then repeating over and over again in sort of another group of muscle fibers. And this is how your muscles work together to contract. Okay, so I'm taking you through this slowly. If you have any questions, yes. So surrounding this muscle fiber, and it's not really clear on this particular diagram, there is a plasma membrane. It's sitting, it is an actual cell, and the cell has all these different proteins in it, and we're just seeing how they're arranged together. And you can think of it as an elongated cell, but all cells have a membrane around it. That membrane is known as a sarcolemma. Okay. So if we see this again, another way to take a look at this, either in cross-section, if we were to cut through in cross-section, this is what it would look like, or we can look at it longitudinally from the side. We would see that these sarcomeres, here is one sarcomere, another sarcomere is another group of actin and myosin, and all of these put together create these striations. This is why your skeletal muscle is striated, these bands of actin and myosin. The sarcomere is a repeated structure over and over again in this skeletal muscle. Any questions about this so far? So right now we've looked at actin, myosin, forming the basic unit, which is this sarcomere. Here is one sarcomere. Here is another sarcomere, and you can see that they're connected together. So if this sarcomere here, for example, were to contract, this sarcomere here would be pulled along with it or contract along with it. Any questions before I go on? So I need you to start thinking a little bit about some of these different myofilaments. And to be honest, this is pure cell biology. So there's a lot of things that hopefully you'll be able to spend some time thinking about, memorizing, going back and putting together. So one of those thin filaments, thin myofilaments, the other one being myosin, and I'll come back and talk about that in a moment. We have two of these actin filaments kind of winding together in a helix. So if you go back to that previous diagram, this this actin filament that you see here, it's not just one actin. You'll actually see two of them interwoven together as a double helix in this actin myofilament. So this is, again, capable of binding to myosin. But are your muscles contracted all the time? Your muscles aren't contracted right now, right? You need a way to be able to turn this on and to turn this off. Okay, you might as well just take the whole stack. Yep. So actin wants to bind myosin. Actin binding to myosin causes this sarcomere to contract. But we know that's not happening all the time. There has to be a way to regulate this. So I want you to remember this because it's really important. So we have this actin filament. It wants to bind myosin. And sitting on top of this actin filament is another molecule known as tropomyosin. And tropomyosin is sort of like having a braid within a weave. Okay, so you have like this weave, and then you have a braid sitting within it. It's another protein. It is not actin. It is another protein. Okay, so don't know if we have a diagram for this coming up, but if you were to take a look, and on this diagram, it's not on there, but I'm hoping that there is another diagram coming up, and I'm pretty sure that there is. So this is sitting on top of actin. It's piggybacking actin. Actin is there on making these cross bridges or trying to make cross bridges. And the other thing that's on top of the actin is this tropomyosin. There's one more thing that piggybacks on top of the tropomyosin as well, and that is troponin or troponin C. Okay, so we have three different layers. So we have on the very top troponin sitting on tropomyosin, which is then sitting on actin. It sounds really, really complicated. And this troponin actually consists of three different subunits. One subunit is there to bind to actin. Another one is there to bind to tropomyosin. And the third part of this troponin, the troponin C, it binds to calcium. Okay, so tropomyosin is connecting, troponin is connecting to tropomyosin, actin, and then also to calcium. Any questions about this before we go on? It's very important that you remember this. It's part of the actin filament. So let me ask you, because I think that it's something, if you understand it, it's really easy, and hopefully it'll be really easy for you in 2 weeks' time. Is troponin found on the thick or on the thin myofilament? So which one is it, thick or thin? It's thin because it's associating with actin, and actin is a thin myofilament. Okay? And we need both of these because this is really what regulates the binding between actin as well as myosin, which is what we need for those sarcomeres to start contracting. So the thick myofilament is this myosin that we looked at earlier. And myosin has two different regions associated with it. It has a rod portion, and then it has sort of a golf club-like portion, which is called the myosin head. So the golf club shaft, for example, they interact with each other, and they form this thick filament in between, and I'll show you this on diagram. And these myosin heads, in theory, can bind to actin. They have a very, very high affinity to bind with actin. Okay, and the other thing to make note of is that these myosin heads also have ATPase activity. So they can bind ATP, they can break down ATP, they want to bind actin, but there has to be a way to stop it from binding actin. Okay, and we're going to get through this mechanism in the next few minutes. Any questions about this before we go on? Okay, so we're going and reviewing some of the things that we've already talked a little bit about. So this is one structural unit that is involved in contraction. It's repeated over and over again in a muscle fiber. This is called the sarcomere. Okay. This sarcomere consists of both thin myofilaments and thick microfilaments, actin versus myosin. If we were to take a look at actin, this is the actin that I just told you a little bit about. There are two double helices of actin molecules wound together in purple. And then sitting on top of that is this tropomyosin that you see here, sort of in this light blue green, sitting on top of the actin. And those small little red dots that you see is the troponin C. So it binds actin as well as tropomyosin, and theoretically it will also bind calcium, which is not being shown here in this particular diagram. The other thick filaments that we see are these myosin molecules that have this capacity to interact with each other through this rod-like domain. And then they also have this head-like domain that you see here that allows and wants to bind to actin. So you can see they're in very close proximity to actin. They want to be able to bind it. As soon as they bind, the muscle, the sarcomere, starts to contract. And that's not happening all the time. You are controlling this contraction. So the reason why this doesn't contract automatically by itself is because these molecules, normally at rest, are preventing actin and the myosin heads from interacting. Okay? Any questions about that before we begin?
Could you repeat that one more time?
Which part? The whole thing?
The last sentence.
I can't remember what I said in the last sentence.
You said the
The reason why it doesn't contract.
So normally at rest now, when you're just sitting there, your muscles aren't always contracting, and the sarcomeres aren't moving together and causing contractions to occur. And the reason why is, even though we have this myosin, which has a very, very high affinity, its normal state is to want to bind to actin and cause contractions. But that doesn't happen normally, because these molecules that are sitting on top of actin prevent myosin from binding. And we're going to try to figure out how to get rid of these molecules that we see on here to allow actin and myosin to interact. I think I just said it in a slightly different way, but hopefully that makes sense to you. Yes?
You know how you said the sarcomere consists of one actin and one myosin? You know how you said the sarcomere consists of one actin and one
Yeah, in this particular cartoon, it's one actin, one myosin. But in fact, there are many of these in one sarcomere.
In the picture, like at the top,
Yep.
there's like 4 actins.
Yeah, again, this is the sarcomere just refers to the end of one part of the sarcomere and the other. It doesn't refer to just these 4 molecules here. It actually goes farther down if you want to think of the diagram extending downwards. Okay, we'll actually get into these Z lines, Z discs, and everything else that marks the end of one sarcomere and the beginning of another. Any other questions before we go on? Okay, and this gets actually into your question. So how do we know what makes up a sarcomere? If they repeat over and over again, how do we know where one starts and one begins? So all of these different filamentous molecules, the actin, myosin, and everything else, but primarily actin, they all attach at what is known as the Z disc. That's the ends of where we have the sarcomere. So it's sort of shaped like a Z or a zigzag, and these Z lines or Z discs are where all of these actin molecules attach. And in fact, they attach to this really large protein. It's the largest protein that we have in the body. It is on the order of mega bases. And this titin filament anchors at the Z disc the actin molecules. If we were to take a look at the striated appearance of actin and myosin, you'd actually see that there are gaps in between the thick and thin filaments. And on a diagram like this one, you can actually see that there is a thick band here, and then there's a lighter band, and then another thick band. And when we go back and start seeing which proteins are in these different regions that I just showed you on that previous slide, we can start identifying them sort of as I bands, A bands, etc. And some of these bands change as the muscle contracts. So for example, the A bands, which I'd like you to make sure that you're circling off, the A bands are really what we see under a microscope there because of just the thick filaments, okay? The M line, which is something I'm going to use in a few moments, is something that marks the middle of what is known as the H zone. And this is where we have filaments that hold myosin in place and allows us to see, is this muscle actually contracting? And I'm going to show you this in the next few slides. So you don't need to memorize all of this diagram or all of the points on this particular slide. You just need to know the the A band, the M line, the Z disc as sort of the outline of what a sarcomere is, because that helps to define what we're going to see a little bit later on. So I told you that you need to keep clear the thin filaments, the thick filaments that make up these muscle fibrils or muscle fibrils. And the other thing that I asked you to make sure that you were aware of as we go through this is how do we actually get muscles to contract when we get nerves to start firing. So I know you guys all remember everything from your last term test, right? Yeah? Okay. So let's say that you have a in the motor cortex, which is found in what part of the brain? Yeah, so it's found in the precentral gyrus. So you have this idea, you've done the motor planning in the forebrain, it sent a signal to the precentral gyrus, a group of neurons in the motor cortex, decides that I want to lift up this recorder. For you to do that, it has sent a signal down to the spinal cord near where the groups of neurons that control arm movement, probably in the cervical to thoracic region of your spinal cord, which have sent out through the ventral horn this motor neuron that is innervating the muscles within my arm. So how does that actually translate into a muscle contraction? So this is that synapse that we talked about before. We're calling it a special name. It's a neuromuscular junction. It's really no different. Here is that motor neuron coming in, is making synapses onto the synaptic membrane, which we're going to call the sarcolemma. This is that postsynaptic membrane of the muscle fibers, that sarcolemma that I asked you to remember earlier on. And it works exactly the same as we've seen with when an action potential comes down, there are vesicles that get released. The neurotransmitter crosses this space and it will bind to receptors here on this postsynaptic motor end plate, otherwise known as the sarcolemma. Any questions about this before we get into more detail that I need you to know? No? Okay. So this gets into how does this all work? And this next few slides is really all about understanding how these muscle filaments slide outside of vision and phototransduction and how you see light in your photoreceptors. This is the best biochemical cascade where we know the most about how the molecules that are involved in muscle contraction contribute to physiological process. So ultimately, actin and myosin have to interact. It's almost inevitable, right? It's like you know for sure that LeBron James is going to be meeting Stephen Curry in the finals of the NBA. It's like a given. Sorry to the Raptors fans out there. I'm a Raptors fan too, but let's be honest. It's inevitable. It's going to happen no matter what you think is going to happen. Actin and myosin are going to interact. They're going to shorten. They're going to slide over each other. The actin and myosin themselves actually don't change their length, okay? It's just that the space in between them is growing shorter and shorter, or is decreasing in length. And I'll show you this in the next diagram. And it's this shortening of the sarcomeres. Remember, it's an end to end. The ends are being pushed in together. And imagine all of the ends in your muscle contracting together. That's what you're seeing when you see your biceps contracting. I wish I was much more ripped or cut or whatever you guys call it now, to be able to show you like a bicep muscle contraction as these things are shortening in length. And imagine this is all these separate units shortening, causing this big muscle to start to contract. During relaxation, these sarcomeres start to lengthen again. So it's the shortening and lengthening that allow for tension to be built up. And we're going to see how this works in a few moments. So this gets back into, this is the basic structure of a striated muscle. This is skeletal muscle. It's striated because we have this repeating pattern of thick microfilaments and then these thin microfilaments around it. If we were to take a look at it in a real micrograph, this is what it would look like. So we have these bands and these striations that we have of light and dark and light and dark again, repeat over and over. And it's because at rest, they're sitting here like this, and we can actually identify separate regions. So for example, we have regions that only identify where we have myosin, and this would be the A band, only with the myosin, and this is that dark line that you see here. There is no overlap with any actin filaments at all in this band here, which is the A band. And this allows us to see what is happening at rest, okay? And it's just for us to be able to see what happens during muscle contraction. Now imagine somehow this muscle was stimulated. Like let's say you put an electrical current on it, or let's say the muscle actually contracted because you thought of it contracting. Now that same muscle, nothing else has changed. This is the same field of view. Everything else is the same. What do you notice about this muscle on the bottom? What do you notice? So the Z discs, those things that were out here, have moved inward like this, right? Do you all agree on that? So hopefully you do, that these discs that were out here, these Z discs that are connecting these sarcomeres, this is one sarcomeric unit. These Z discs have moved together. It has indeed contracted. In fact, one of the things that has happened is that space that we saw here, that little A band that we saw here earlier, has disappeared. Haven't shrunk at all. The myosin filaments haven't shrunk at all, but the space in between them has lessened, has decreased as the two ends have squished together. The myosin is still exactly the same size as it was before, and so is the actin. It's just moved closer together. Have my fingers changed their overall length? They have not. I've simply moved them closer together, and that's what you're seeing here. The actin and myosin represent my fingers. My fingers are not changing length, but the reason why it looks like they're getting shorter is because the space in between them is disappearing as the actin and myosin form these cross bridges. Is that clear for everyone? So I'm hoping that you'll remember that the ends of these sarcomeres are marked by these Z bands. And this is why we talked about these Z bands. We talk about this A line, or part of this H band, disappearing as it shrinks. And again, I want you to know some of the details that I covered in that previous slide. So again, if we're to take a look at a muscle fiber, and we go back to the same principle that we had earlier on, and we stuck in an electrode, just like a neuron, this muscle fiber, I'm not talking about the nerve that supplies the muscle. We're talking about the actual muscle fiber, okay? The muscle fiber, not the nerve to the muscle, but the muscle fiber itself has a voltage change across it. It is already having a resting membrane potential. We can excite this muscle, just like we can excite a neuron, okay? And that's the most important part that you should be taking away from all of this. I don't really care about the potassium. It's exactly the same as a neuron. There's more potassium inside than outside. Potassium leaks out. There's a sodium-potassium pump, but it's really the same as a neuron. It's not very much different. If you're a really good physiologists, you'll note that the resting membrane potential is a little lower. It's at minus 85. It's not at the minus 70 that we looked at earlier. But for the most part, it looks almost identical to what we see with the neuron. And this again gets into, well, if it kind of has a resting membrane potential, can we change that membrane potential across this muscle fiber using another neuron? And this gets into these neurotransmitters. We've already talked about it. At the neuromuscular junction, when we have a motor neuron trying to excite a muscle tissue, a skeletal muscle, it is always releasing acetylcholine. The particular receptor for this acetylcholine is the nicotinic receptor that I'll show you in the next diagram. But this is the really important part, that it's always releasing acetylcholine. Motor neuron is going to release acetylcholine. Just like we talked about in the nervous system, we have acetylcholinesterase within this neuromuscular junction. It is a degrading enzyme. We don't want your muscles to continue to contract after you have sent the signal in. We need to get rid of the acetylcholine, and we have acetylcholinesterase here as well. So we're going to start putting all of this together now in the next few slides. So if you're feeling lost, let me know. I'll slow down. It's important that you know the next few slides really well, because it's a really important mechanism. And I told you at the outset, you need to know from skeletal muscle, you need to know what are the molecules that are involved. So far we have actin, myosin, tropomyosin, troponin C, or troponin. And I told you also you needed to understand how a nerve excites the muscle, and now we're getting into that. And then you need to be able to talk about how the cross bridges, how the sarcomeres actually shorten. So now we're into the second part. How do you actually excite this particular muscle? This is the muscle cell that we want to be exciting. An action potential has come down from the motor neuron. It causes the influx of calcium. Calcium binds to these neurotransmitter vesicles. It causes exocytosis, and the release of acetylcholine crosses this neuromuscular junction, or NMJ, which is just like a normal synapse. Acetylcholine will then bind to the receptors on the muscle membrane. And what was this muscle membrane called? Sarcolemma. The sarcolemma then has these nicotinic receptors that are found here, which when acetylcholine binds, opens up a gate that allows sodium to flow through. And as these sodium channels open and as sodium flows through, this muscle cell, this muscle fiber, now starts to depolarize, just like we saw in the neuron. Questions about this before we go on? And again, we don't want acetylcholine to stay out here too long, and so we have acetylcholinesterase, which will break down acetylcholine to acetyl as well as to choline. Some of it gets taken back up, some of it diffuses away, and we have a very short regulated signal. So if we continue with this, eventually that acetylcholine will unbind. The ligand-gated channels close. The acetylcholinesterase starts to break these things down. Some of it gets transported back. But we've caused this depolarization in this particular membrane, in this muscle fiber. Okay, is that clear for everyone? An action potential has come down from a neuron, and that action potential from a neuron has excited this muscle membrane. Maybe? Maybe. Okay. So ultimately, why does it do that? Why do we actually need this really convoluted mechanism? So there are a couple of things that are important about this whole membrane that you see running down here, depolarizing, and it spreads. It's a spreading wave of depolarization. It might start here, and it will work its way all the way down to the end of this muscle fiber. So this involves this sarcolemma, this membrane that's found on the outside. This outside pinkish covering represents the plasma membrane. Here are all of those different thick and thin filaments just waiting to start making these cross bridges. They can't right now. They're blocked by which two molecules? Tropomyosin is one. What's the other one sitting on top of tropomyosin? troponin. So it's blocked. They can't move right now. They want to be able to slide over top of each other. Those Z bands want to start contracting. And we're just waiting for one thing to happen. We're waiting for calcium. And this is really why we have this action potential coming in, causing this wave of excitation traveling down this muscle fiber. Because somehow we have to get calcium onto that troponin. If we can do that, then actin and myosin will do everything else for us. So where does that calcium come from? That's one thing you should be asking yourselves over the next few slides. And then ultimately, how does that calcium get released from this action potential that I'm going to be covering in the next few minutes? So if you're to take a look at the structure of what this all looks like, this is the sarcolemma. Built into this, we have these invaginations, which are known as transverse T-tubules. And these T-tubules are really membrane that actually work its way into the muscle fiber. Okay, so it's like having stairwells that go down a few levels. You're all in the same building, you're all on the same floor, but if you were to go up and down different floors, you would use these T-tubules to allow the depolarization not just to stay on the surface, but also go down deep into the muscle fiber. Because if it's only up here at the top, all these fibers down here in the middle wouldn't get depolarized. Another thing that you can see associated with this is that we have a particular structure known as the sarcoplasmic reticulum. Do any of you know what the sarcoplasmic reticulum is? It's like the endoplasmic reticulum of the muscle cells. So what do you store in the endoplasmic reticulum normally? Really? Normally? Yeah, so it's used for things like protein transport and protein export. In this particular case, the sarcoplasmic reticulum is the store of calcium. This is the major store for calcium in skeletal muscle. So just again, to put this in context, the sarcoplasmic reticulum is right beside where these T-tubules are. The T-tubules carry depolarization that we need. Calcium is just waiting to be activated, and it's being stored in this sarcoplasmic reticulum. Okay, so if we're putting all of this together now, and we want this interaction to occur, right? We want to eventually see LeBron James meeting up with Stephen Curry in the finals. It's one of those inevitable things. The only thing that we have to do is get past, and I guess OKC, right? So how do we get rid of OKC? We'll call OKC tropomyosin, and we'll call, I don't like the Toronto Raptors being called troponin, but let's say the Toronto Raptors are like troponin. Just how do we get rid of that interaction? So this action potential that we saw earlier on, that was produced at that neuromuscular junction, has spread along the outside of the membrane. It's activated nicotinic receptors and has caused this sodium to flow in along this membrane. We also saw that there were these invaginations, these T-tubules, that are the membranes that go down deep inside these muscle fibers that we saw a little bit earlier on. And sitting right beside them is this sarcoplasmic reticulum. So one of the things that happens is that as that spreading wave of depolarization passes, and as it works its way down these T-tubules, that depolarization activates a voltage-gated calcium channel that is found inside the sarcoplasmic reticulum. And look at the location of where the sarcoplasmic reticulum is. It's right beside these actin and myosin filaments. So as the depolarization activates these voltage-gated or voltage-dependent calcium channels in the sarcoplasmic reticulum to open, that calcium that's been stored in there, that now has a voltage-gated calcium channel opened up, now flows out because the concentration gradient allows it to flow outward. That means that now calcium is now inundating and flooding all of this actin and myosin sarcomere that is related around it. Is that clear for everyone before I go on? Set in motion by the muscle, by the nerve, releasing acetylcholine, has activated sodium or the influx of sodium through the nicotinic receptors, and this in turn has now opened up the sarcoplasmic reticulum calcium stores to release calcium into the region of the sarcomere. Okay or not okay? All right. So why is all of that important? Normally, these myosin heads that want to attach to the actin, it's blocked. It just can't get to it because the tropomyosin and the troponin that's sitting on top of it prevents actin and myosin from binding. It's like a physical blockade so that it can't bind to these purple regions that you see here. As soon as calcium binds to troponin, the troponin that sits on top of the tropomyosin causes a conformational shift, and it rotates the molecules off of actin. They don't completely disappear, they're still attached, but what happens is the calcium binding causes troponin to change its orientation, and the troponin changes the tropomyosin that it's attached to change its orientation, and as a result, the whole complex moves off of actin, and now actin and myosin have this capacity to interact and form these interactions that we're calling cross bridges. Is that okay for everyone? It only can take place when calcium is there. Any questions before we go on? Hopefully that's somewhat clear for you. And again, this is that calcium ion being released, causing the troponin, tropomyosin to move off. Normally it's been blocking this interaction. Now it's being moved off. These active sites are now exposed, allowing these the tropomyosin, the myosin and the actin to bind and these cross bridges to form. Any questions about this? You need to know this mechanism. Anything that you're not 100% clear about. So one of the things that happens is if calcium is there, and only if calcium is there, this allows for actin and myosin to start binding. I told you at the very outset, about 20 slides ago, that the myosin, the thick filament, the thick myofilament that you see here actually is an ATPase. Do you guys remember that at all? It has ATPase activity. So now this is ATP, is just about ready to be broken down. The myosin has ATPase activity. It is now bound to actin, and as it binds to actin, it is going to start breaking this ATP down and using the energy of that ATP to actually cause that myosin head to start flexing. And as it moves, and you can imagine a whole bunch of these things moving at the same time, it is going to cause the actin to slide over top of the myosin. Okay, so as the ATP starts to be broken down, it causes this conformational shift in the myosin head. That stored energy is used to move the actin along this way. In other words, the myosin is moving actin and it's pushing it and contracting it together in what is known as the power stroke. And then as soon as that happens, another ATP molecule, because now it has used up all the ATP, another ATP molecule will then come in and attach itself to the myosin head, which is what you're seeing here. That interaction of ATP with the myosin head causes the actin and myosin to break off, okay? So this then allows it to reset. The actin has already been pushed along. ATP has come in, it's reset, and now it will bind again, and it will repeat doing this over and over again. The same myosin head will continue to interact and push along myosin as long as there's calcium around and as long as there's ATP around. Okay? So we need, for your muscles to work well, you need both calcium as well as ATP. Okay, and this process again is being repeated over and over again. Any questions about this before we go on? Any questions? Yes.
So one ATP will then a new ATP will unbind it. So you just
So the ATP is there to begin the process. It will be broken down, allow for the myosin to bind to actin. It then causes a power stroke of the myosin head. Once that power stroke is completed, there's no more ATP around. The ADP moves off, allows for ATP, another ATP to bind. That resets the myosin head and allows it to repeat the whole process again. Bind actin and continue on. Questions? Will you guys remember this for May the 30th? No, not 100% sure about that. I hope you will. Again, this is for your own information. This is a good summary of everything that we've talked about. So I told you that you need to understand this mechanism up here, have an action potential causing nicotinic receptors to be activated, causing this sarcolemma to depolarize, the release of calcium. You need to know the molecules that are involved in this sarcomere that you see here, and you need to understand the whole basis, the molecular basis of this actin-myosin sliding filament hypothesis. And it's probably the most famous biochemical hypothesis that still exists today. There's no one that's disproven this hypothesis. Any questions about this before we go on? This is a great summary slide. If you understand everything on this slide, and here's a good way for you to test yourself. When you go back, and I told you that you need to know 3 things from this section on skeletal muscle. You need to know the different components of the sarcomere. You need to understand this excitation contraction coupling, and then you need to know the mechanisms that are involved in cross bridge formation and sliding filament hypothesis. If you can understand everything on this slide, then you will get perfect on this section. Okay? If you have any questions after reviewing on the weekend, then you should come and talk to me before Wednesday's class to make sure that you understand it. And I think now is actually a good time to take a break. And if you have not picked up your test, you can go ahead and do so now. We'll take a break for about 10 minutes. Anyone else? I'm gonna be posting the music video for you, but hopefully you should. Anyone else? No, I'm not sure. I don't think we do. And yeah, so muscle cramps can actually occur for a number of reasons. I think one of the summary slides at the very end talk about a number of different things like fibromyalgia, muscle cramps. I think is in there as well. But there are a number of reasons. One of this is that build up of lactic acid, build up of phosphate. So if you are doing breakdowns,
So the inorganic phosphate. So it has to do with nerves, or it's just a phosphate breakdown.
it's just yeah, it's kind of like muscle fatigue.
Yeah. So for that class, I'm just
Just before the
So how about if you see this calcium to go
Okay, folks, if you don't mind, we have a lot to cover over the next little while, so and I want to make sure that we cover everything that you need to know for your term test. So if you don't mind, I'm going to get going. So we have just been talking about muscle contraction and how it is coupled with nerve cell activity or motor neuron activity. In terms of muscle relaxation, the whole process is just basically reversed. The calcium that has started this process, that is bound to troponin, and has moved off tropomyosin, exposing the actin and myosin. The calcium is just removed. As soon as that calcium is removed, it moves back into the sarcoplasmic reticulum through active transport mechanisms. In other words, this is another reason why you need ATP. It's also ATP dependent. And then calcium, again, allows the removal of calcium allows for the reestablishment of the troponin, tropomyosin, covering on the actin and myosin to block those binding sites. So if we're taking a look at the whole skeletal muscle cell physiology, there's a couple of really interesting points that I'm going to point out to you. So if, for example, we were to apply a stimulus, so we're not talking about a nerve actually causing this, but if you were to inject current into the muscle cell directly, instead of seeing an immediate response, there is a little bit of a lag. So there is a lag or latent phase between excitation as well as muscle contraction, and we measure muscle contraction by the amount of tension or power that is generated by the muscle as it starts contracting. So even if we start stimulating here, and this is where we apply the stimulus, electrical activity, we don't actually see the muscle doing anything because of this lag response. Eventually, though, as it produces an action potential, more and more of those muscle fibers get recruited, more and more actin, myosin cross bridges form. The more cross bridges that form, the more power you are able to generate, the stronger the contraction is, in other words. And eventually, this contraction phase stops and this relaxation phase begins. And this gets into this idea. You have a whole group of different muscles, and these are things that I want you to be able to point out and pick out in the next few slides. So again, skeletal muscle histology and biology is what we covered in the first sort of hour. In the next little hour, as we go through this, I need you to be able to pick out things related to motor units, what a motor unit is, the different types of muscle fibers as we start talking about them. And again, you're going to get about 20-ish questions. So we've already looked at all the sections that we've already had. We're already talking about 8 questions from that. We're going to be talking about another 4 to 5 questions related to motor units, properties of different types of muscles as we start to go through these. So a motor unit is one single motor neuron that we looked at earlier, and all of the different muscle fibers that get innervated with it. So it's like a hand going down. How many different muscle fibers can that hand sort of innervate? That would be one motor unit. So my arm is that single motor neuron. My fingers represent all the different muscle fibers that it touches. And for motor unit numbers, larger muscles, like really big biceps muscles, the postural muscles, etc., have motor units that will make contact with many different muscle fibers. And then small muscles, like in your fingertips, for example, that are involved in delicate movements, contain motor units that have smaller numbers of muscle fibers that get activated, right? You don't need a lot of muscles being contracted for your fingers, but you need a lot of muscles being contracted in your biceps. So there's a difference in large muscles and small muscles. True, just like for the nervous system, we talked about that all or none. So you either generate an action potential or not. So if you are below the threshold stimulus, because a muscle fiber itself has electrical activity associated with it, if you are below that threshold stimulus, you can't change the polarization state across that membrane. You will not generate an action potential that spreads along the muscle fiber, opening up sarcoplasmic reticulum and calcium release, and you will have no contraction. No action potential, no spread, no contraction. If you reach the threshold stimulus, then an action potential spreads along the sarcolemma and it will cause this contraction. So again, just like with the nerve cell, the stronger the threshold or supra-threshold, action potentials are still going to be generated, but we reach a maximum state of contraction. Like just applying more action potentials doesn't make your muscle contract any more forcefully than it is capable of doing. So if we were to take a look, the strength of contraction is graded. So the more motor units that we can activate. So if we activate just a few, then just a few of my muscle fibers in my arm are going to contract. I won't be able to actually, if you've ever tried like lifting something, you have to recruit more and more muscle fibers as you're lifting something heavier. You're seeing the recruitment of more and more of these muscle fibers up until a certain maximum. So imagine for a second, so I started off with a small number of muscle fibers being recruited, more and more get recruited, but I can't actually lift much more than that. That's sort of like, I hate to say it, but that's about the limit of my muscle strength. So no matter how much more motor units I try to recruit, I can't actually lift anymore. I can't actually go beyond this supramaximal stimulus. I can't actually produce a stronger contraction. I can't produce something that's going to be like way up here by having more motor units being activated. So what we're seeing here is the summation of more and more of these muscle motor units being recruited, okay? So this phenomenon that I'd like you to make sure that you are aware of. So I told you that these muscle, the strength of contraction can be graded. So it's can go from small to big, in other words. So we can see a gradual increase in the tension that is generated or the muscle strength that is being generated. This gradation is something that we call trepe, and this trepe is a graded response, okay? Generally speaking, when we talk about these graded responses, as we try to recruit more and more muscle fibers to this, each contraction gets bigger and bigger over time, and then we reach our maximum where we can't actually generate any more strength or any more tension. And one of the reasons why we think this sorts of things occur is that every stimulus that we apply, so here is one stimulus, it produces this response, another stimulus produces this response, the third stimulus produces this other graded response, and then when we start stimulating more, we get the same sort of response. The reason why we get trepe when we apply continual stimulation or multiple stimulation is we think that that calcium, so it hasn't had a chance to completely disappear. Those actin-myosin cross bridges are still there, and another wave comes in and activates a little bit more of them, causing this trepe response. This would be a really bad way for your muscles to work, though. So your muscles normally don't, unless you have a condition, your muscles don't usually twitch. Like if you've ever seen, if you've been like walking all day long and you look at your leg and you see this muscle twitching, have any of you ever seen that? Sort of your muscle, you're not actually doing anything, and your leg is actually like the muscles in your leg are actually twitching. That's not a really good way for your muscles to work. For you to be able to do anything useful, these little twitches where your muscles are turning on and off, a long hike and you're kind of resting from that, these little twitches are not something that we really want to have. Your muscles can't do anything if it's like twitching off and on. I couldn't lift anything if it's twitching on then twitching off. And so when we are looking at these little muscle twitches here, it is something that is not a physiological response that we want our muscles to normally have. If we start increasing the number of action potentials coming down that motor neuron, we should be recruiting more and more of these muscle fibers. But for us to actually get complete muscle contraction, we need to have what is known as complete tetanus. We have to have the muscles contracting in such a way that all the muscle fibers at the same time are fully contracting. These little twitches of going off and on and off, on and off, on and off, on and off. If we increase the frequency of action potentials, then we get that trepe that we talked about earlier. So there's this graded response. If we increase it even more, a higher frequency, more action potentials, it's closer in time. It's that summation that we looked at earlier. And then eventually we reach the right frequency where all the muscle fibers together at the same time are contracting together as a concert, in concert. And it'd be like asking all of you at the same time to shout, Raptors will win, right? And if I say 1, 2, 3, if everyone was on target, we would reach this sort of complete tetanus. Everyone at the same time would say Raptors will win, but more likely someone will start off saying Raptors will win. Some of you won't say it, and it'll look kind of like this response here. But we don't want that kind of response when we talk about muscle, because one of the things that we want with muscle is to be able to generate power through this active tension. So if we need to lift something, we need to have tension and contraction force within the muscle to be able to do this. If you are stretching out your muscle, or you have a muscle that is already crumpled and it's already sort of in a contracted state. So a cramped muscle, I think you were asking about. If you have a muscle cramp, it doesn't work as well as it normally should because it's already kind of contracting and spasmodic. So if we were to take a look at this, there's actually an optimal length that your muscles have to be in order for them to generate the most power that they possibly can. Okay, and we won't worry about passive tension versus total tension. I just want you to be familiar with this idea of power or tension, which is related to contraction of these muscles, and the state of the muscle, whether your muscle is stretched, whether it's already contracted, makes a difference in terms of what it is capable of doing. So this is sort of what we're looking at. This is the optimal length of muscle. This optimal length of muscle is if we were to stimulate this muscle, it would be able to generate this much tension up here at the top. And this would be if it's in the optimal state. It's not compressed, nor is it elongated. And that reason why we can produce this maximal stretch is that the number of cross bridges that can form, the number of actin, myosin, cross bridges that can form here is actually at the optimal length. If we have a muscle that is already contracted, it's already shrunk, then we see that the number of cross bridges, we can't actually get much more. We've already got a lot in this particular state. And if we look at the state where we have this muscle stretched out and we want to generate power from a stretched out muscle, then you can see that the number of cross bridges that are possible for you to form decreases as well. So it's all about muscle strength, muscle tension is always equivalent to the number of cross bridges that can form. So the more cross bridges that you can form, the more the muscle can contract. The more the muscle can contract, the more strength or tension you'll be able to generate, which is what this diagram is showing. Do you have any questions about this before we go on? No? So again, there are a number of different types of muscle contractions. There are contractions where they are isometric. So if you're standing, your muscles aren't actually, you don't actually see your back muscles contracting or anything else, but they're there and they're working. They're there to keep you upright. So I want you to make sure that you know that postural muscles, particularly in your back, are there to give you an isometric type of muscle contraction. An isotonic type of muscle contraction is where you have a change in length, but the tension may be constant. And it's this type of thing that is used in resistance training. Okay, so if you're working out and doing resistance training, we're using this isotonic type of muscle contraction. And I don't need you to memorize anything on this particular slide. We've gone through really quickly each of these different points. On this particular slide, I want you to be able to point out examples of what muscle groups are involved in isometric contraction, what sorts of things might be useful for isotonic contraction on that previous slide. You should be able to give me a good working definition that you can apply on a multiple choice test question related to trepe, which is a graded muscle contraction that we saw earlier on. And again, we looked at different types of summation and why we need tetanus in the muscle in this particular scenario, and the tetanus being the most important thing that you should be taking away. So I told you another thing that I need you to make sure that you know from this particular section are the different muscle fiber types, okay? And you need to know this slide because this slide is actually a really important one in terms of biology. There's a lot of controversy related to this particular slide as well. So you have a number of different muscle fiber types. And these are different muscle groups, for example. We have a type of muscle which is called the slow twitch oxidative muscle fiber type. These tend to contract more slowly. Um, uh, uh, worry too much about fatiguing. They are fatigue resistant. In other words, you can contract your muscles for these types of muscles for a very, very long period of time, and they're not going to wear out. You're not going to get tired from these muscles contracting. These generally tend to be smaller in diameter than the fast twitch type. They have a better blood supply. There is more mitochondria in the slow twitch oxidative type. Again, the name oxidative suggests that they use a lot of mitochondria. They also contain a lot of the protein known as myoglobin that delivers oxygen to the different muscle groups. A good example of this are the postural muscles. So we have already talked about the postural muscles in terms of different types of contraction on that previous diagram. And again, we see a lot of these slow twitch oxidative types in the lower limbs, in your legs in particular, to be able to keep your posture and in the upright condition. We use these slow twitch oxidative type. And again, if you are thinking about this, because these are fatiguing less quickly, and fatiguing mean like if you were to hold a weight in your arm for about 5 minutes. Like let's say you were holding up a 20-pound weight in your arm. After a few minutes, depending on your physical conditioning, you're going to start feeling muscle fatigue. It's going to start hurting. These muscles, though, are fatigue resistant. Like you can stand around for long periods of time. In theory, if you were really good, you'd be able to stand like all day, and your muscles would be pretty much okay. Like you'll be tired at the end of it, and when you go to rest, you'll feel tired. But while you're actually doing that activity, you'll be fine. These fast twitch, though, are a little bit different. These respond incredibly quickly. If you're an Olympic sprinter, for example, you're using a lot of your fast twitch muscles. You have lots of these fast twitch muscles, especially in your lower limbs. They're the ones that respond. As soon as you say go, these are the ones that start contracting. They can break down ATP much more quickly. There is a lower blood supply. So if you've ever seen a sprinter after they do their race, in order for them to be able to recover, what are the things that they do? So you've just sprinted. You've just used up glycolysis. You've produced all of these end byproducts of glycolysis. It's in your muscle. What do you see sprinters do after? Sorry, they walk a little bit. Sure, they walk. Anything else? They take glucose powder. Okay, I don't know. Maybe some sprinters do. They drink something sugary. Okay, sure. Yes. They breathe heavily. Yes, okay. Also, all good. The other thing that you'll often see them do, especially during between training intervals, because they don't have a big blood supply, because these muscles use and break down ATP really quickly, there's no blood supply to remove the metabolites. You'll often see them sort of getting a massage done on their lower legs to try to get the breakdown products of metabolism back into the circulation through the lymphatic system through that. So they don't have as many mitochondria, and the mitochondria that they do have tend to be much smaller than the slow twitch type. Again, we typically see this in the lower limbs of sprinters. In your biceps, for example, these are the fast twitch muscles. As soon as you need to use your biceps, they go, and these are the ones that you'll see most often. And again, because of the lack of myoglobin, myoglobin is what gives dark meat its color, especially in chicken. If you ask for dark meat, you're eating lots and lots of myoglobin in that meat. If you are looking at white meat, it doesn't have that myoglobin, and it's lighter in color as a result. So there are, again, two different types: fast twitch oxidative and fast twitch glycolytic. The ones that use the most energy, most powerful types, the ones that you use for weightlifting, sprinting, and everything else, are the glycolytic forms, and the oxidative are a little less powerful. Okay, so most muscle groups or most muscles in the body have both. It really depends on the individual, how much training they've done, etc. It also depends a lot on the type of exercise. I told you that that last slide was really controversial. There were a lot of studies that tried to show and demonstrate that you could actually, do you have to be born as an Olympic sprinter, or if you were born with a whole bunch of these slow oxidative type, can you convert your slow oxidative muscle groups? If you're not an Olympic sprinter, you're not genetically predisposed, your mom and dad are not Olympic sprinters, can you become, through training, and convert some of your muscles into the fast type? And there's a lot of controversy about this. We all know that we can, through exercise, change the size of the muscle fibers. But can you actually change the molecular mechanisms? Can you change the number of mitochondria, the myoglobin that's found in it? That's been a lot more controversial. We know, for example, that if you are exercising, generally your muscles get bigger, right? Unless you're doing the wrong kind of exercise and your muscles are getting smaller. I don't think most of you have seen that happen. But for the most part, they hypertrophy. And the muscle hypertrophy that you see is a direct effect of this exercise. It is because you are increasing the number of myofibrils. You're also increasing the nuclei, because some of the satellite cells that we didn't go into detail increase the numbers, and they fuse their nuclei together. So the number of cells is increasing. Again, as you start increasing in strength, it's likely also due to a better coordination of the different muscle groups, an increase in the production of these metabolic enzymes. You're also going to be, for some of these muscle groups, increasing the blood supply to those muscles, or better circulation to the muscles. And of course, you're also going to be reducing the amount of fat associated with this. Again, in atrophy, where you have a decrease in muscle size, so you might be not using your muscles for long periods of time. You might have a neurodegenerative condition where the nerves don't get to your muscles. You can't contract your muscles anymore. So things like ALS, for example, motor neuron diseases, you'll see those individuals in wheelchairs. Their muscles are almost completely gone because they've atrophied and decreased in size because they weren't using it. If you are, again, using your muscles during exercise, both the metabolic rate at which the muscles are using energy as well as the heat production increases. The metabolic rate tends to stay high after exercise is done in your muscles. This is primarily due to oxygen debt. So some of you were talking a little bit about what do sprinters do after they finish running. Some of them will breathe heavily. Some of them will kind of lie down and try to recover a little bit. And again, this excess heat that is continuing after this exercise will eventually be lost through vasodilation, increases in the blood vessel diameter, as well as sweating. In certain cases, you have lost control of your hypothalamus, and you have just finished exercising, and you have started to overheat. In those rare instances, you can actually have someone on a hot day after exercising starting to shiver because they have increased the contraction even more of these different muscle fibers. It's very rare, but it does happen in some athletes after exercise is complete. So you're gonna have to tell me, because it's up to you. I believe in fair choice wherever possible in a course. Do you guys like biochemistry, or not such big fans of biochemistry? You love biochemistry. You really love biochemistry. Yes. Okay. So this is for your own information then, the next few slides. One of the things that is really important for muscle contraction, there are two things. We've already talked about these two things. One is you need ATP. ATP. Why do your muscles need ATP? Yes, energy is good, but it's not a great answer. It's a good answer. So the myosin is an ATPase. It allows for the cross bridges to move. And we need to be able to replenish the ATP as much as possible. There's another place that we saw ATP though. What else that uses ATP? do you remember we have calcium? We need to get rid of calcium. We need to put it back into the sarcoplasmic reticulum. Does it require ATP or not? It does require ATP, and so this ATP actually is there to put the calcium back into the sarcoplasmic reticulum. So some of you will also know that ATP can be produced in a number of different ways. So we can do it through oxidative phosphorylation or aerobic respiration. We can produce it through the glycolysis or anaerobic respiration. We can also use it or create ATP, very small amounts, through the creatinine phosphatase reaction. And all of this requires oxygen, especially for aerobic respiration, and we tend to go into oxygen debt during, or oxygen deficit during periods of exercise. So again, we won't worry about the different mechanisms that are involved in ATP production, but if you are interested in this, you can go through how ATP works. It's just for your own information. We won't go into it in any detail. The creatinine phosphate, you might actually be into weightlifting, or any of you lifters? Do you lift? Do you even lift? No one here lifts. Okay. I think we should start doing that. Some of you might be taking supplements to help you in the recovery phase, like creatine supplements, like the creatine phosphate. Again, for your own information, so we'll skip over the biochemistry of ATP, other than knowing the importance of ATP in the whole process. Okay? On this before we go on, or anything that you want to go back and double check whether or not you have to know. So the ATP section, the biochemistry, the reaction pathways, the molecules, you really don't have to know. The reason why we need ATP though in muscle, you do have to know. Okay, this gets into muscle fatigue. So this is the decreased capacity for the muscles to actually work, decreased or reduced efficiency of performance of the muscles. There can be a number of different reasons for muscle fatigue. Some of it is psychological. That's really complex. Muscular, this could result from not having enough ATP around. Then you have this muscle fatigue. It could be for synaptic regions. You may not have enough acetylcholine around, no acetylcholine, no nicotinic receptor activation, no depolarization, no calcium, no muscle contraction. And that's another reason for muscle fatigue as well. This gets back into some of the other reasons why we worry about things like calcium. So if you were looking at the normal physiological way in which the muscles contract, and this particular case known as rigor mortis, we need to have calcium and ATP. This is normally required for physiological contraction of your muscles. And in rigor mortis, one of the things that happens, if you've ever seen someone who's passed away fairly recently, if you've ever watched shows like CSI where they're trying to time the death of an individual, they will actually be looking for rigor mortis, because within a few hours after death, after someone stopped breathing and has not taken in any oxygen, all the muscles actually go rigid. You can't actually move their muscle groups really easily. You can't lift their limbs easily. It feels like they're kind of fighting back against you, even though you know that they are dead. And this rigor mortis happens because when you die, you don't have ATP. That ATP is there normally to put calcium back into the sarcoplasmic reticulum. Without that ATP, that sarcoplasmic reticulum actually leaks calcium back out the other way. You have all this calcium, you have all these cross bridges forming. In fact, you form the maximal number of cross bridges that you possibly can. And then eventually this rigor mortis will disappear. The actin and myosin filaments themselves will start to disintegrate as the body continues to decompose. And within about half a day or so, the rigor mortis kind of goes away, the actin and myosin filaments disappear, and the rigor mortis, the body starts becoming flaccid. Then there are some other physiological, pathological changes like bloating, the accumulation or leakage of body fluids. So this is, the timing of death is really something that can be measured through rigor mortis. So we've talked really briefly about oxygen deficit and recovery, and this again relates back to ATP production. If you don't have oxygen to use in aerobic respiration, you can't do oxidative phosphorylation, you can't form 36 molecules of ATP for every glucose molecule that you're breaking down. This oxygen deficit becomes really important after exercise. And you need to be able to get that oxygen going to produce that ATP. And this oxygen consumption, again, is something that's important after exercise as well. Okay, any questions on this before we go on? So this is where we're going to switch gears just a little bit. At the outset, I told you that you should know various things about skeletal muscle. So you should know the molecules in skeletal muscle that are involved in contraction. You should know about the excitation mechanisms that are involved. You should know about the sliding filament hypothesis. I told you that you should know about the different muscle fiber types, oxidative, fast twitch, slow oxidative types. And I told you that you should also know a little bit about the ways in which muscles contract, things like trepe, tetanus, etc. That's all skeletal muscle. Now we're going to switch gears, and I told you at the outset, for smooth muscle, I want you to concentrate again on the mechanisms that underlie smooth muscle. The calcium stores are a little bit different. The ways they contract are a little bit different. But also, in addition to that, there are different smooth muscle types, and you need to be able to differentiate those. If you can do all of those three things, identify the different molecules that are associated with smooth muscle, how those different components work together to cause smooth muscle contraction, and the different types of smooth muscle, then you'll be all set for this section on your term test. Okay, so we do not see striations in smooth muscle. That is why it gets its name. When you see smooth muscle, the fibers don't have striations. We don't see the actin myosin fibers repeating. We don't see sarcomeres repeating. We do have smaller fibers, and we have spindle-shaped fibers, as well as a central nucleus. Remember, the nucleus is on the periphery, on the edges in skeletal muscle. Here, the nucleus is where we normally see it inside the cell. It's a central nucleus. Smooth muscle, we don't have that cross-bridge formation like we did with skeletal muscle. We actually have more actin than we do myosin. In addition to this, we also have this other structure that I'd like you to make sure that you always associate with smooth muscle when you're thinking about muscle, these calveoli, which are these indentations in the sarcolemma. So it's still a muscle. We still call the plasma membrane a sarcolemma in muscle, even if it is smooth muscle. We're not 100% sure. We don't have T-tubules in smooth muscle, but we do have these calveoli, and it might be that these indentations, which we call calveoli, work like T-tubules. So if you're asked, have T-tubules, but they do have calveoli that look a lot like these T-tubules. We don't have these Z-discs either. Z-discs are unique to those skeletal muscles. Those are the endpoints where we see which molecule associated with Z-discs. Titin. So the titin, the largest protein ever found, titin is found at the Z-discs embedding the actin and myosin, the actin filaments in there. We don't have Z-discs like we do in skeletal muscle, and we have non-contractile intermediate filament proteins instead that anchor these actin and myosin filaments. We still require calcium. The source of calcium is a little bit different, and calcium in this case is not binding to troponin. Calcium in this case is binding to calmodulin. The mechanism, in other words, is different. When calcium is activated in smooth muscle, calcium will bind to calmodulin, which will then activate myosin kinase, and this myosin kinase allows for actin and myosin to form cross bridges. It is a very different mechanism, even though actin and myosin are still the same molecules. Again, in order for this to stop working, we need to have a phosphatase come in and remove the phosphate groups that were put on with that myosin kinase. So if we were to take a look at smooth muscle, we would see that it has this very unique type of structure. We see a central nucleus. Instead of having the nucleus pushed off to the periphery and having this elongated muscle cell that we saw earlier, this is more or less a normal looking muscle cell or a cell. The nucleus is in this centralized region, and we see all of these different myosin filaments in between these actin filaments that run crisscross throughout the entire cell. When calcium enters into the cell, and it causes the activation of myosin kinase, it will cause some of these myosin units to interact with actin to cause shrinkage of the cell just like before, but the mechanism for its activation is different. It's not an ATPase dependent fashion, it is a kinase dependent fashion. So we're going to go through this again in some more detail. So if you have any questions along the way, please let me know, because it is important that you know how this works. So again, we're primarily interested in this calcium channel. I'm going to skip through the hormone here and the hormonal activation of smooth muscle contraction. I'm more interested in the calcium channel that we're looking at here. Smooth muscle doesn't have a nerve. Okay, so smooth muscle can get activated by hormones. It can be activated by a number of factors. It doesn't have a nerve to activate it, which is why in this case we're starting off with a hormone. But I'm going to skip through the G protein coupled mechanism and jump directly down to this calcium channel. So somehow, either through hormonal activation or through some other depolarization effect, you can open up this calcium channel that you see here. Calcium will then flow into the cell. So it's not coming in from the sarcoplasmic reticulum. It's extracellular calcium in this case. And this calcium channel that has now been opened, whether it's through a hormone and a G protein or through depolarization, whatever it is, this open calcium channel allows calcium to flow in. Calcium will then bind to the calmodulin subunit. This calcium calmodulin subunit then activates myosin kinase, and this myosin kinase acts as the ATPase, not the myosin head itself directly, and this allows the myosin to attach to the actin in a slightly different way. In order for it to detach, we need this myosin phosphatase to come in and remove the phosphate group and allow this to reset itself. So very, very different. I needed to keep the mechanism for skeletal versus smooth muscle clear. You guys are probably thinking you're going to get this on your term test. And you're right, you are going to get this on your term test. It is a basic mechanism that you should know about smooth muscle contraction versus skeletal muscle contraction. Okay, so again, calcium and calmodulin activate this myosin kinase. It attaches to a phosphate group to the myosin head and allows this cross-bridge formation to occur. So some elements look similar, but the mechanism for the actin-myosin cross-bridge formation completely different, and activation and deactivation of this cross-bridge formation is very different as well. So again, you know you're going to get a question on it. Make sure that you know the differences between skeletal versus smooth muscle. The source of calcium is one. Sarcoplasmic reticulum doesn't exist in smooth muscle. It's all extracellular. There's a calcium channel that is involved here. We're not worried about the G protein coupled hormonal mechanisms, but you should be aware of all of these downstream effects that you see here. So this is sort of that one piece that I asked you to make sure that you know. Do you have any questions on this one piece from smooth muscle that you need to know from this section? Any questions at all? The other thing that I asked you to make sure that you knew were the different types of smooth muscle cells. So we can identify smooth muscle cells or different smooth muscle cells by the physiology, by the way they work, as well as some specific markers for these. So we have what are known as visceral or unitary smooth muscle, and these are cells that are found together in sheets. So they're not isolated cells, they're found together as sheets. They function and contract together as a functional unit. So the whole thing contracts together. They contain a number of different gap junctions. So as soon as one group of smooth muscle cells contract, that calcium passes all the way through and it causes a wave of contraction. These are often autorhythmic, so they start up and stop all by themselves, just like the heart is autorhythmic. The visceral cells are also autorhythmic as well. So an example of where we might find these visceral or unitary smooth muscle cells would be in your digestive system gut wall. We would see lots of these visceral types. This is in contrast to the multi-unit type, where we have groups of cells that are completely independent of each other. So they can also be in sheets, like in the blood vessels. They can form bundles, and again, this is sort of where it's a group of cells, and this could be in the iris of the eye, or they can be single cells like the capsule of the spleen that we'll eventually be coming to later on in the course. These are sort of the other type, the multi-unit type. Any questions about this or what you need to know before we go on? Okay, so some of the other things to know, and then we'll take a break after about 2 slides, are some of the electrical properties of smooth muscle. So smooth muscle, if you were to take a look at it, have these slow waves. So this is on sort of the a smooth muscle. If you were measuring from it, you would actually see that it starts to depolarize and then it starts to repolarize. And this depolarization, this spontaneous depolarization and repolarization, this wave of depolarization that you see down here on the bottom, is caused by the spontaneous diffusion inward of both sodium and calcium into the cell. This is not like a neuron. It's not all or none. It's not an action potential. So as sodium and calcium work its way in, it starts to depolarize, and then it will start to leave and it will start to repolarize. If we are looking at some smooth muscles though, some of these smooth muscles actually may start generating action potential. So they'll reach threshold, and if they reach threshold and they're still depolarized, they may start firing action potentials. In some other cell types, you might actually see where the action potentials don't come back down, but stay in this prolonged state. And we're going to come back and see how some of these smooth muscles act like pacemaker cells, where they set the rhythm of contractions. Can you think of a place in your body, muscles that contract on a rhythmic basis? Your heart is not smooth muscle, but it is a rhythmic basis. Smooth muscle. Stomach? Intestines? Good answer. Agree with, we have 2 choices, stomach or intestines. Stomach? Intestines? Any other answers? Any other guesses? So even if you're not eating, from the top of your digestive tract, which includes your esophagus, all the way down to the anal canal, and we'll get to this in the digestive system, your entire smooth muscle within your digestive tract contracts on the order of once every 30 some odd minutes or so. There's a wave of depolarization that goes down. It's followed by a contraction all the way down. Even if you're not eating, it speeds up if you're eating, but this is an example of where you have pacemakers that cause regulated forms of contraction. And some smooth muscles will have this autorhythmic contraction that we'll see within the gut, and it tends to respond to things like stretch in addition to having this auto rhythmicity, and we see a lot of this, especially within the gut, that the amplitude of contraction remains fairly constant. The only thing that changes is the muscle length itself tends to. So some of the things that I want you to make sure that you know about the regulation of smooth muscle, we've already seen that the autonomic nervous system controls a lot of the smooth muscle functions. The neurotransmitters that can regulate smooth muscle are things like acetylcholine as well as norepinephrine. Just when you thought you didn't need to know norepinephrine or norepinephrine or acetylcholine ever again, it's back. Do you think you need to know it for your term test? Probably. That's a pretty good bet. Do you guys remember what the receptor types that we're talking about here? What is the receptor that interacts with noradrenaline? It's adrenergic, that's correct. And acetylcholine, you have two choices, nicotinic, muscarinic. In this case, when we're talking about smooth muscle, do you know which one we're talking about? Go all the way back here, and it's a big hint for you. So this is a hormone and is activating this cascade. Which of those different receptor types work in a similar fashion? Is nicotinic a G protein coupled receptor? No, we just talked about it with skeletal muscle. It opens up a channel, sodium flows in. Muscarinic is a G protein, adrenergic is a G protein. When we talk about smooth muscles, we're talking about G protein coupled receptors that will modify the activity of smooth muscle. Any questions before we go on to the next section? No, so I think it's actually a good time to take a break. Then we'll get into cardiac muscle, and then we'll get into the cardiovascular system. Okay, folks, if you don't mind, I'm going to get going. We'll try to get through as many slides as possible to make sure that you are ready over the weekend to start studying for your next term test. It's about how well your class did in terms of the average class mark. So I can tell you that you were over 72%. And getting pressure from the other class, though, to kind of keep things more similar to their class. I'm trying to resist that a little bit, so I'll let you know by next week. My goal is anyways not to make it cumulative as much as possible, but if it is cumulative at all, which I'm hoping it isn't, I will reduce it dramatically so that it's maybe 1 or 2 questions at most, and I'll be very clear on what you need to study for that. So you don't have to panic about it or anything else. I want to be able to keep my promise as much as I possibly can, in keeping with like what people at Ryerson are telling me I should be doing with these different courses. So let's get into cardiac muscle and then finish off this module on muscle and then get directly into the cardiovascular system. Okay, so the cardiac muscle, again, we're going to go through this in more detail in the next section immediately after these next few slides, but this gets into the cardiac muscle kind of being an intermediate type of muscle. So we only find it in the heart. Smooth muscle is found in a number of different places in the body. Skeletal muscle is found in a number of different places in the body, but cardiac muscle is only found in the heart. We don't find it anywhere else. It has striations, but they're not ordered striations. We don't see the regular repeating sarcomeric subunits in the same way that we do in skeletal muscle. The cells, again, have one nucleus. They also have some unique structures like intercalated discs. They also contain gap junctions. What do you think gap junctions would be useful for? We've talked about gap junctions before. Sorry? So it's like an electrical synapse where we need to have the entire structure depolarizing at the same time. It makes sense that you don't want little bits of the heart depolarizing. You want everything depolarizing at the same time, and these gap junctions help to do that. The heart also has autorhythmic cells. They have what's known as pacemakers. Your gut has a pacemaker. It contracts like every one time every 30 minutes. Your heart has a pacemaker associated with it. In fact, there are 2 pacemaker cells within your heart that give it the regular 72 beats per minute that we're used to, and that's the autorhythmicity of the cardiac muscle cells. The action potentials, the depolarization and the contraction that's associated with cardiac muscles are much, much longer than they are for the nerves, and they also have a longer refractory period. Once you have contracted a heart muscle, in other words, you can't get it to contract again, and you can't get it to fire an action potential again because it has a longer refractory period. We also need calcium here to cause cardiac muscles to depolarize and contract as well. So do we need calcium for all the muscle groups that we've talked about today? Yes, we definitely do. And we'll go through this again in some more details in a few moments. As you get older for skeletal muscle, unless you're taking growth hormone, your skeletal muscle mass is going to decrease. Your muscles, no matter what you do, even if you're exercising, you're still going to have diminished muscle mass. Part of that is because as you get older, you don't produce as much growth hormone. If you take growth hormone supplements, your muscle mass tends to stay relatively intact. There is increased time for your muscle to contract in terms of a response to nervous stimuli. So in other words, you're not as fast as reacting. You're not as fast with your reaction times as you were when you were younger. There is less ability for you to have sustained muscular activity. If you do have sustained muscular activity, it takes longer to recover. And of course, loss of muscle fibers, decreases in capillary density. All of these are associated with the aging muscular system. Again, this is for your own information. I'm not going to go over this in detail. I think you were asking about this earlier in terms of cramping or fibromyalgia. There are many different types of muscle disorders. If you're interested in them, you should be able to, based on what we've talked about, figure out why it causes the types of things that they do. However, I'm not going to be testing you on these separately. It's for your own information if you're interested at all in applying some of what we've learned to these muscular system disorders. The only thing I'll touch on that you don't have to know is this here, DMD. Have any of you heard of DMD, Duchenne's muscular dystrophy? Do, the disorder looks like? So this is usually a really severe disorder depending on the type of Duchenne's muscular dystrophy you have. Your muscles atrophy. You turn 13, 14 years of age, your muscles, you've been developing normally, but then your muscles start to atrophy. You're not actually able to stand up well either. So you have this very characteristic rolling on the floor because your muscles and your legs are so weak, you actually have to climb up to stand upright. And this form of Duchenne's muscular dystrophy is a really severe disorder. But in Toronto, we actually have a lot of researchers who are working on trying to find cures for Duchenne's muscular dystrophy. If any of you are in what is that, biomedical sciences, have you heard of CRISPR-Cas9? CRISPR editing? So genome editing, where you can actually edit the genome either in an adult or in a developing embryo. Right now it's not possible to, well, it is possible, but there's a blockade on embryo testing. But in adults, for Duchenne's muscular dystrophy, using CRISPR-Cas9 and a technique called exon skipping, they've actually been able to cure Duchenne's muscular dystrophy in different animal models. And so there's a lot of hope that even though some of these different muscle disorders are very, very severe, that with biotechnology, we're actually able to reverse a lot of these different phenotypes. And this was actually the coolest story that I shared with my students over at the University of Toronto. It was one of those remarkable things that happens once in a while in science. Do you guys know the different kinds of journals that there are for publishing scientific articles? Have you heard of Nature? Have you heard of the journal called Science? So are these good journals or not so good journals? So these are the top rated journals. And on the very last day of the last month of 2015, in a back-to-back-to-back issue of Science magazine, because they were making a statement, the editors of Science actually published 3 papers from 3 separate high-powered labs that showed that you can actually cure Duchenne's muscular dystrophy using CRISPR-Cas9. The reason why they did that was to show that in 2016, that we should be using these technologies to try to answer and cure some of these disorders that up until 2015 were thought to be lethal. And the hope is there that maybe CRISPR-Cas9 and other genome editing techniques might actually allow us to cure things like Duchenne's muscular dystrophy. And again, it's a remarkable series of papers. If you're interested in doing a review article that you haven't picked one out yet, those are the papers that I might draw your attention to. They're fairly easy to understand, very powerful techniques, and you'll understand something that is at the leading edge of science, and I'd be happy to actually go through the papers with you. So we're going to skip over a couple of things and get directly into the cardiovascular system. So for your next term test, I need to get through the remainder of the cardiovascular system next Wednesday, as well as the respiratory system next Wednesday. And those will be the two, this lecture and that lecture will be on your term test. So the heart has a number of different functions that we need to understand to look at some of the different functions of the heart. So we need to be able to use the heart to generate what is known as blood pressure. So you'll understand blood pressure a little bit later on. It has to be able to drive blood into both the lungs, which is the pulmonary system, as well as into the systemic circulation. We need to make sure that blood is always going in one direction, and so we need specific valves to make sure that blood is going in one way and only in one direction. And we need to be able to regulate the blood supply. So if we're to take a look, and again, all of this is information that we'll be covering. You don't have to memorize this slide now. It's just an overview slide for you. We actually have 2 hearts in one. We actually have a heart that supplies the lungs, and this is the right side of the heart. And then the left side of the heart has that blood coming back from the lungs, and then it pushes it out and drives the oxygenated blood coming from the lungs that you see, this oxygen coming in. We breathe off CO2, and then it drives it through the rest of the circulation, and then it comes back through veins back to the right side of the heart where it completes this circuit. All the CO2 is blown off, and then new oxygen is put back in place. So in terms of the relative gross anatomy of the heart, your heart is roughly the same size as your own fist. So it's relative to you. It's not like my fist is the same size of heart that all of you have. It's your own fist that you should be looking at. It has an apex, a pointy end, in other words, associated with it, or a blunt, rounded end, but we call it the apex, which is more pointy than the base, which is the flat part. And it's found in the thoracic cavity near the central core, which is known as the mediastinum. And this is important. So we're going to come to this, I think, today. But this mediastinum and the location of the heart, the way that the heart is rotated within the thoracic cavity is actually really, really important. This is actually crucial to what we now know about modern medicine. So the next time you go to your doctor, think a little bit about what they're doing when they're using their stethoscope, right? So if you've ever gone in recently to have your to visit your GP, your doctor, his or her stethoscope, puts the stethoscope either on the chest or on your back, right? What else do? They just kind of leave it there? Your doctor actually knows what they're doing, right? So when they're kind of moving it around, so when they're kind of moving it from one location on your chest to another location, it's not that they can't find your heart. It's actually because of the mediastinum, because of the orientation of the heart, they're actually checking for your valves opening and closing, and they can only do that because of the way your heart is oriented in the thoracic cavity. And it's not that he or she doesn't know where things are. They're listening for specific locations on your chest and on your back to be able to hear these different valves that I mentioned at the very outset. So you have this heart sort of surrounded in the thoracic cavity with the lungs around it. We're going to go through the different orientations of the heart in a few moments. I'm going to tell you that I'm not going to ask you to know or go into a lot of detail about the coverings of the heart. Normally, when you have an organ, and your heart is an organ, it's a really big organ, if it's moving around an awful lot, you don't want it rubbing up against anything else within a cavity. So your lungs are in their own sac, and they have a pleural sac, and your heart has another sac that makes sure that it's not rubbing up against anything as well. And so it has its own pericardial sac. And there are other layers that surround the heart as well. There is a fibrous layer, there is a serous layer that produces the actual fluid. Anytime you see serous, it means that it is producing fluid. So if you see that in future anatomy courses or physiology courses, serous meaning that at some point that tissue is going to be producing fluid, and this is where the fluid gets into the pericardial sac. Again, we won't worry about all of the anatomical details here. It's just to give you an idea that your heart is moving around robustly. All of these different fibrous and pericardial layers are there to protect your heart as it's moving around. Again, this is the lining that we just talked about. It has to be protected in this double membrane sac. So we have the fibrous and then the serous pericardium. And in between, we have this fluid that protects the heart as it's beating and as it's moving around in the thoracic cavity. So this is a slide that I want you to spend a little bit of time on. Not everything on this slide, nor do you have to worry about having a diagram on this. And I told you that we don't really need to worry about all of the different layers of the pericardium, the sac-like covering on the outside of the heart. But if we're looking at the heart wall, the last layer, the serous layer, makes up one of the walls of the heart. And the outer layer is the epicardial layer that you see up here, sort of in this yellowish color. Then we actually have the muscle fibers itself. So this is the muscle cells, the cardiac muscle cells. You can see the blue nuclei in between the muscle cells. You can see some gap junctions there. And this is that myocardium in this middle layer. And this is what is actually going to do all the contracting. Then there's this endocardial layer outlined for you sort of in this bluish color. And this inner layer is where we might have some extra modifications. We might have these trabeculae carnae, which are muscular ridges that might be found if we're looking at a particular chamber of the heart known as the ventricle. So this slide, in terms of just the anatomy as I've outlined it for you, you don't have to, again, worry about any diagrams or anything else happening to it. The heart is a muscle, and just like the skeletal muscle and having blood vessels supplying it, this is actually somewhat relevant and somewhat important for all of you. This is the coronary circulation. So we talk about the systemic circulation, how blood goes from the heart, the left side of the heart, to the rest of your body. We talk about the pulmonary circulation, of how blood goes from the right side of the heart and gets pushed through the lung before returning to the left. But the heart itself needs its own blood supply. It needs it to be able to function normally. When something goes wrong and one of these different coronary arteries gets blocked, then you have a heart attack. You feel pain in your heart because those visceral receptors are responding to metabolites being released by cardiac muscle cells as they're dying off. No oxygen, no ATP. That cardiac cell is dying. It releases factors that the visceral nerves actually can detect. Again, I don't want to go into a lot of details about the overall anatomy, but I do want you to know a little bit about the coronary circulation. So we have a right coronary artery, which will then form a right marginal artery, and you don't have to worry where it's going. And we also have this interventricular artery as well. All of these are important because any one of these that get blocked will result in a different type of heart attack depending on which of these different coronary arteries get blocked. The left coronary artery, which is on this side over here and is behind this particular pulmonary trunk, has a number of branches as well. And I want you to be, again, aware of these just in terms of being able to identify them as part of the coronary circulation. The circumflex artery is one of those arteries that comes off the left coronary. The marginal artery, just like the right marginal artery, comes off the coronary. And then there's also an interventricular. The one difference here is this circumflex artery. And again, when you're having a heart attack, it's more likely that you're going to have the right coronary artery being blocked, but you can also block the circumflex. You can also see blockade of the coronary. And all it takes is just a little bit of fat deposition in a blood vessel that supplies the heart, blocks the blood vessel, so there's no blood supply. It blocks it here. This artery gets blocked. The entire right ventricle gets no oxygen, and it hurts. And it hurts like you are having heartburn, but that heartburn persists for long periods of time. And if that doesn't clear up, that heart muscle is going to die. Your heart isn't going to function normally, and you're going to start getting edema. So you're going to get fluid buildup in your lungs, and that itself is not a really pleasant thing as well. We don't really get blockade of the veins. And again, this slide I don't need you to memorize at all, so you can just strike this one off. It's just to give you an idea that with the arteries, we also have to be talking about the venous system. And again, there are large vessels like the cardiac vein, the coronary sinus, and a number of smaller veins. But we usually don't worry about these veins very often because they don't tend to get blocked. And hopefully by the end of today's lecture, you'll understand why arteries get blocked and veins don't, right? Your veins can get damaged. Your veins can get damaged in a number of ways, but blockade by fat or atherosclerosis or plaque deposition only happens in arteries. It doesn't happen at all in veins. Any questions about what you need to know before we go on? No? Okay, so I told you earlier it's a lot like having two separate hearts. We have two side as well as a right side. And when we look at the right side, we have a smaller chamber at the top, which we call the atrium, which then empties to the right ventricle. And this is part of the pulmonary trunk, which means it's supplying the or going into the pulmonary system, which is your lungs. So this is where deoxygenated blood full of carbon dioxide is going to go into your lungs through the right atrium, through the right ventricle, into the pulmonary circulation, in toward your lungs. It will eventually come back into your heart through the left atrium that is receiving blood from the pulmonary veins, and will eventually pump it through the left ventricle, and it will then go to the rest of your body through the aorta. Okay, and again, I don't want to make it any more complicated than that. Between the left and right, because we need the left side of the heart to be completely separate and distinct from the right side of the heart, there is a wall which we call a septum, and this interatrial septum is a wall that separates the left and right side of the atria, and we also have a wall between the ventricles as well. We need to make sure that the blood going to the lungs is kept separate from the blood that's going to the body. And this is sort of the overall diagram. Usually for whatever reasons, the oxygenated part, the left ventricle that is pushing blood or the left side of the heart that's pushing blood into the rest of your body is depicted as red, and the deoxygenated blood that's pumping blood pumping from the right side of the heart into the lungs is usually depicted as blue, but all of you will know that those different colors don't really exist in blood. It's usually darker red versus brighter red, and we'll talk a little bit about that in the next lecture. So I told you that there were a number of different valves that were important in this whole process, and this is an important slide for you to make sure that you know. So we have valves that separate the left atrium from the left ventricle, and we have a valve that separates the right atrium from the right ventricle. I told you maybe about an hour ago that blood has to be flowing in a unidirectional manner. It has to be going in one way. Blood goes one way. If it comes back from the ventricle back up into the aorta, that's a real problem. That's actually a form of regurgitation that actually decreases your health status. And so we need to make sure that blood is always going only from atria to ventricles, whether it's left side or right side, always atria to ventricles, never backwards from ventricle to atria. And the only way to do that is to have a valve that allows it to go from the atria to the ventricle, and as soon as the ventricle is filled, it closes. And so to do this, we have these AV valves, which are these cusp-like muscle attachments attached by those chordae tendineae that I mentioned to you earlier. The right side of the heart actually has 3 cusps or cups if you were to look at it anatomically. And so between the right atria and right ventricle, to prevent backflow, we have this tricuspid atrioventricular valve. The left side has 2 cusps or only 2 cup-like openings, which is why it's called the bicuspid or the mitral valve. Do you have any questions about this? And you need to be able to keep these straight. Left side has 2, the right side has 3. Any questions? No questions? Can I ask you a question? Why mitral? The bicuspid makes sense. Tricuspid 3, bicuspid 2, mitral, why? See, this is why you should ask questions, so I don't ask you questions. Any ideas? So for any of you who are in the Roman Catholic faith, if you know the bishop's hat, do you know what the bishop's hat is called? No one's Roman Catholic? Have you ever heard of a bishop's mitre? They have a hat that sort of has this sort of V-like shape to it. And this bishop's miter, which has two prongs going up and then the dome on top, that double miter is why it's called the mitral valve. It's named after the bishop's miter. And this is, again, an AV valve that I want you to make sure that you are aware of, because blood always has to be flowing in a unidirectional way or a unidirectional manner. If it leaves the ventricle, it shouldn't come back into the ventricle. So from the left aorta, the left ventricle to the aorta, it shouldn't come back from the aorta back into the ventricle. It has to be unidirectional, atria, ventricle, then aorta. To prevent backflow there, we have the semilunar valves that are associated with the right and left side of the heart, and these semilunar valves are there to also prevent backflow as well. So we have atrioventricular valves, semilunar valves. All of these are there to maintain this one-way flow so that we don't get blood going back from the from the ventricle back up into the atria, and it's always sort of this unidirectional flow in one way. And this is shown for you here in this diagram. You don't have to memorize it. Hopefully by the end of this section you'll have gotten the most of it. If you are really a diagrammatic or a visual person and you prefer having it schematically and it's easier for you to memorize it, we just went through earlier, this text here, this is actually a diagrammatic way. Right atrium blood flows through the tricuspid valve into the right ventricle, and this is just a different way to give you the same information that was on the previous slides, and it also introduces you to these semilunar valves that I mentioned to you earlier to prevent backflow from the aorta back into the left ventricle, etc. So it's just a different diagram. If you're a visual person, this might be easier for you. If you just like having text and memorizing from text, then this is the slide that you might want to use. Any questions before we go on? Okay, so we're going to be talking a little bit more about the overall histology of the heart. If we were to strip away all of the different layers of the heart and we were only left with the cartilaginous fibrous rings that were left here, we would be left with these different valves that you see here. And again, it's a very interesting sort of connective tissue that you see in the sort of skeleton of the heart around the atria as well as the ventricles. We'll see a lot of fibrous rings around the valves to provide this support. And it's also a way to prevent electrical activity from jumping between the atria and the ventricles. We need all of the heart to be functioning at the same time, and we don't need it to go atria ventricles. We need atria and ventricles to fire together, and I'll show you how it does that in a few moments. So in terms of going back to the cardiac muscle, and we started this discussion a little bit earlier on, and then we sort of took a little bit of a detour and talked about the anatomy of the heart. So going back to cardiac muscle, we talked about these different cell types that we saw earlier. These are branching cell types. They also contain actin and myosin myofilaments, just like every other cell type that we looked at. Smooth muscle has it, skeletal muscle has it in a very organized fashion. Cardiac muscle has this as well. This has some unique properties associated with it too, though. We only see intercalated discs in cardiac muscle. Don't see it in smooth. We don't see it in skeletal muscle. These are specialized cell-cell contacts. These are where gap junctions allow these different heart cardiomyocytes to interact with each other. They're held together by these structural elements known as desmosomes. Because they're contracting, we need to be able to anchor all these cells together. They're anchored together through these desmosomes, and the gap junctions act like pores between the different cardiac muscle cells. So the gap junctions will allow the action potential to spread really quickly from one cell to its nearby neighbors. And all of the cardiac muscle in the atria and ventricles, because of these gap junctions and because of the intercalated discs, are functioning almost like that smooth muscle. They all work together at the same time. So they're all behaving as a single unit, much like the smooth muscle example that we looked at earlier. Okay, so this is an important slide for you to know as well. So this is one that you might want to get back and start thinking a little bit about. And the next slide gives you a little bit more detail about this as well. I told you when we talked about smooth muscle, and at the very last slide in the muscle section about cardiac muscle, the cardiac muscle has autorhythmicity. In fact, if I were really, really bad, I would take your heart muscle out, your whole heart out. It would still beat by itself. Like it doesn't need nervous system activity or anything else. It naturally, spontaneously would start beating as long as I provided it with nutrients, as long as I kept it in a solution. It would beat as long as the muscle was kept alive. And one of the reasons why it beats is that in the atria that you see over here, um, we have a particular group of cells which are known as the sinoatrial node cells, and these act as pacemakers. And these pacemakers will cause depolarization in a very rhythmic fashion. So they spontaneously depolarize, fire action potentials, then go quiet, spontaneously depolarize, cause action potentials, and then go quiet. And they will also connect to the AV nodes. So they'll go from the sinoatrial down to the AV node, and this AV node itself also has spontaneous pacemaker activity. And the reason why we have two is we can regulate the function of one, and it turns on and off the function of the other. And by coupling this, and because of the way in which these AV node sends fibers out to the rest of the ventricles, it can cause the entire ventricle to actually start depolarizing at the same time. So as soon as the atria here, the ventricles will start to depolarize almost simultaneously because the action potentials get spread out across the entire ventricles. Okay, so both of them at the same time. So there are a number of anatomical fibers that branch off from this. The most important one being the Purkinje fibers that you see here spreading up into the muscle walls of the left and right ventricle. I'll come back and talk about the SA as well as the AV node in a moment. So I don't mean to be disingenuous, but I like asking details because this one is actually really important. Um, Do you see the SA and the AV node in both atria? Yes? No? No. Okay. And it's not inherent in this particular slide here. But which of the two atria do we find the SA and the AV node? Yeah, it's in the right atrium. So make sure that you are aware that if you're looking at the right atrium, one way to identify the right atrium is to be able to detect spontaneous pacemaker activity. We only see the SA and AV nodes in the right atrium. We don't see that in the left atrium. We don't have those specialized cell types on the left. And again, that might be something that's important for you on May the 30th. So this SA node, the sinoatrial node that begins this whole process, are specialized cardiac muscle cells that have autorhythmicity. They generate those spontaneous action potentials. They will then pass these a very short distance onto the AV node, and these AV nodes then spread the action potentials through the rest of the ventricles so that both the atria, when they contract, the ventricles will contract almost simultaneously with them. So again, the Purkinje fibers that I mentioned earlier on being the ones that innervate the rest of the ventricular muscle cells to allow the spread into the ventricles. Any questions about what you need to know here? None of the other branches that are here, you don't need to really worry about those. Okay, so when we've been talking about this cardiac muscle, this is sort of that pacemaker potential that we've been looking at. It does this by itself. It does it in a dish. You can actually dissect out these cells from the right atrium, put them in a dish. They will spontaneously generate these action potentials over and over again in a very rhythmic manner, and it will start this sodium and calcium influx. The voltage dependent calcium channels start to close and repolarize in this downward phase, and it's much like the action potential that we saw earlier. There's some sodium entry, potassium as well, and again, it's sort of this rhythmic fashion that will repeat itself multiple times. And just like the action potential we saw in the nervous system, we also have what is known as a refractory period. There's an absolute refractory period and a relative refractory period as well. Just like the nervous system, cardiac muscle cells, no matter how strong the stimulus is during the absolute refractory period, you cannot get that muscle cell to fire another action potential. The relative one, if it's a really, really strong stimulus, you might be able to elicit a response during the relative refractory period. And the reason why we need these refractory periods is the heart has to be able to contract. We need to give the muscle cells time to be able to contract, and this tetanic contraction, the concerted contraction of all the muscle cells together needs this long absolute refractory period. Just out of curiosity, have any of you ever had this? Because the heart is so electrically active, just like your brain is sitting up here. The electrical activity of your brain, it's hard to measure. Even with an EEG, there's these small alpha, beta waves. Once in a while, you'll see some delta waves. And if you're sleeping, you'll see lots of synchronized activity. But your heart is synchronized. And so we can pick up the electrical activity of all the cardiac muscle cells firing together in a synchronized fashion from the surface of your chest through what's known as an EKG or an electrocardiogram or ECG. Right? EKG is the German form for ECG, and it's all about the electrocardiogram. Have any of you had one done? A couple of you have. So I need you to be aware of a couple of things. On an EKG, we have this very, very characteristic, every last person who has a normal heart has exactly the same features on their EKG. So we always start off with a P wave, and then we have what's known as this QRS complex. And then we have this T wave that you see here, and then it repeats itself. Another P wave, QRS complex, and T wave. Which one is the biggest? What's the highest peak? The QRS complex is the biggest. So let's take a look at this and try to figure this out. So you have this P wave, and this represents the atria depolarizing. Depolarization leads to contraction. Makes some sense. So if you were to go back and take a look at the heart, and again, I'll come back to this, which one has more muscle? So if this is muscle up here sort of in a side profile, do you have more muscle in terms of thickness here or down here in the ventricles? So ventricles are bigger. So let's try to make use of this when we get into this PQRS complex. So the P wave is atrial myocardial cells depolarizing. They're all depolarizing together, which means that we can see this electrical event. And it is a relatively sizable electrical event, but compared to the overall size of the ventricular depolarization that we're measuring with the same electrodes, the ventricular depolarization and repolarization that you're seeing in the T wave, it's always much bigger because it's related to the number of cells that are exhibiting electrical activity. So the P wave, atrial depolarization and contraction, the QRS complex, which is much bigger, is the ventricular contraction and depolarization, and then this T wave is where the repolarization occurs. Clinically, we can actually measure what's known as the PQ interval, the time between the start of the P wave and the beginning of the QRS complex, because it tells us how long that signal takes to cross. We can also measure the QT interval because it tells us about the heart. If your heart is functioning normally, if you've had a heart attack, you don't have as many cardiomyocytes functioning, then you can also affect this QT interval. And again, it's for your own information. These ECGs and again, this is for your own information, can provide you with lots of information related to all sorts of things that can go wrong with your heart. So some of you might be walking around right now, you might have an arrhythmia. Your heart actually doesn't spontaneously have this autorhythmicity on a regular basis. It will start skipping a beat or two once in a while. Or you might have it where you are exercising and you might have what's known as a long QT interval. And this is actually much more common than you think. If you've ever read in the newspaper about a really young individual, like 20, 25 years of age and good physical health, and they've gone out for a run, they sat down and they died of a heart attack, they have an undiagnosed QT interval. You won't know until you have this long QT, and it's a hereditary thing that most of you will not even suspect that you have until that first episode happens to you, or you happen to be really lucky, you go in for an EKG and they see that there's something wrong with your EKG. So these arrhythmias actually play a really big role in health as well as in disease. So this gets into this idea that the heart is two different pumps working together, serving different organ systems, the lungs versus the rest of the body, or the systemic circulation. There is contraction, which we call systole, and then there is relaxation, where the heart fills, called diastole, in the different chambers. So there's a pumping, and then there's a filling. The pumping is the systole, and then the filling is the diastole. Where have you heard similar terms before? Yes. Yeah, so when you check your blood pressure and you read the systolic versus the diastolic blood pressure volumes, it's related in some ways to the systolic pressure in your heart and the diastolic pressure within your heart. So again, we won't go through isometric, isovolumetric contraction, and this gets into some aspects that we might come back to later on, but I'm going to skip through a few of these slides for the time being. So this slide here on isovolumetric contraction, we don't have to worry about, and I'm not going to worry too much about the period of ejection nor the isovolumetric relaxation period. I'm going to talk a little bit about some of the ventricular filling. And this really relates to when you are filling your different vessels. So if you're filling your ventricles, for example, your whole heart actually doesn't have to be involved in cardiac output, like the normal blood supply to the rest of your body. You don't need both atria as well as ventricles working 100%. So most of the ventricular filling actually occurs passively. Like even though we keep telling you that, you know, your atria contract and then it pushes the blood down into the ventricles, and then your ventricles contract and then they push it out into the lungs or into the rest of your body. Your heart fills just fine without the any kind of any kind of assist through the atria actually pushing it through. It's only when you actually need to have this, it's only when you need strong atrial contraction in places like exercise where you actually have to go above sort of that 70% filling. So this is where I told you about the SA node that's found on the right side of the heart. This SA node actually can speed up the rate of contraction of the atria, making them more effective in pushing blood out into the ventricles. So under normal situations, when your ventricles are filling to about 70% capacity and you're just sitting here, that's fine. You don't actually need your atria to really squeeze out every last amount of blood that it possibly can. But when you're exercising and your body is making these high demands on oxygen, your atria actually need to be working. You need the SA node firing more rapidly, increasing the rate of the heart muscle contraction to cause that 300 to 400% increase in the effectiveness of the heart. So this is where the atria are important. In the passive filling, we don't need it. At rest, you don't need the atria involved at all. But in active ventricular filling, where we need the atria, we need to speed things up through this SA node. And again, this is sort of a way to look at it clinically. We won't spend any time going through the different filling events that are going on. I'm going to ask you to go back during the weekend to make sure that you understand the events that are associated with the EKG, what sorts of things are depolarizing and repolarizing in these different events. And I'm just going to take you through the last 3 or 4 slides related to the heart sounds, and this gets into what I was talking to you about earlier. Your heart makes sounds. You guys have all heard the lub-dub sound, right? Yes, kind of. So what do you think actually makes that sound? So without looking at this, blood leaving. So blood hitting like your heart, really hard. So how come you can't, like if I took a stethoscope, your blood is flowing through your arteries in your arms, how come I can't hear the blood there? Because it's silent, it's totally silent. It's still like hitting the sides of the walls. I can tell you it's hitting the sides of the walls because I can measure the pressure. When you put a blood pressure cuff, it's hitting the sides of the walls of your blood vessels so hard that it raises the mercury at about 120 millimeters. That's your systolic. How come I can't hear it? Is it really the blood hitting the heart really, really hard as it's rushing in? It's like I'm trying to get out of class because I can't stand it and I got to go home and watch the basketball game today. I hit the door really hard. That's what you're telling me. What is the lub-dub sound? Yes. So it's really the sound of the valves opening and closing. And this AV valve opening and closing at the beginning of this ventricular pumping or systole as it's pushing blood out is that first heart sound, the lub sound. So I need you to make sure that you know this. It's an AV sound, the AV valve that causes this lub. The second heart sound that you hear on a stethoscope when you're listening to the heart is this dub sound, or in this case, dup sound. And this is the closing of the aortic and pulmonary semilunar valves when the heart is filling during ventricular diastole. And this usually lasts a little bit longer. Occasionally, and this is both depending on your age, it can be a really serious thing or it can mean absolutely nothing. There could be a third heart sound that's caused by turbulent blood flow into the ventricles, and it might be detected near that first third of diastole. So I need you to know what accounts for the lub and the dub sound. The third heart sound, which in some ways sounds like a heart murmur, if you hear the regurgitation or turbulent blood flow, most of you have it. Doesn't really make any difference. You're perfectly fine. You can exercise perfectly fine. For whatever reasons, there is turbulent blood flow and you sometimes hear it. So when your doctor is checking for your heart, when you're going in for a checkup, and when they're moving the stethoscope around on your chest or on your back, they're actually looking for specific anatomical markers that allow them to hear both the lub and the dub sound. And again, you don't have to worry about being able to isolate this, but I wanted you to have a physiological context. Why does your doctor move that stethoscope around? It's because they're trying to find the AV valve and the semilunar and pulmonary valves to make sure that your heart is actually working the way that it should. So again, make sure that you memorize things on this slide. If it doesn't make any sense to you, maybe refer back to this slide down the road. And I think this might be actually a good time to maybe wrap things up. Before you disappear, just a reminder, there is no class on Monday. You should really, to be honest, at the cottage this weekend. If you're going away this weekend, before you go away or while you're at the cottage, spend an hour or two going over today's lecture. It'll save you a lot of time next weekend, okay? All right, have a great Victoria Day weekend. I will see you all next Wednesday.